Hi all, welcome back. Today's discussion will be on the chapter How do organisms reproduce in plants. We'll be covering this chapter in two parts. The part 1 will include a short introduction on reproduction and its types, and we'll be looking into detail about asexual reproduction in plants. Second part will be dealing with sexual reproduction in plants, which will be our next video. So, what is asexual reproduction? Asexual reproduction involves only one parent, and there is no formation and fusion of gametes. The young ones will be formed almost identical to each other as well as to the parent cell. In unicellular organisms, asexual reproduction can be taken place in three different ways: binary fission, multiple fission, or budding. Let's talk about each in detail. Now let's talk about binary fission. Binary fission is usually seen in bacteria and protozoa like amoeba. In these, the pseudopodia is first withdrawn. Later, the nucleus of the parent cell divides, and then the cytoplasm divides. Finally, the result will be formation of two daughter cells. This is the basic process behind binary fission. The division of cytoplasm is termed as cytokinesis, and the division of nucleus is termed as karyokinesis. This binary fission occurs during highly favorable conditions. Let's now talk about budding. This is usually seen in hydra. The parent hydra will develop a bud at its lower end. This grows in size and finally breaks off to live independently. This is almost similar to the budding in unicellular organism, except the difference that the bud develops in the lower end. Spore formation. These are seen in Rhizopus, a fungus. Rhizopus body is made up of a thread-like structure called hyphae. The erect hyphae bear sporangia, inside which reproductive structures called spores are formed. These spores are asexually reproducing bodies which have a thick protective wall. They are produced during unfavorable times and help to tide over the unfavorable environmental conditions. When the spores fall on a suitable medium, each one forms a new individual. This tissue culture is a technique of developing new plants from cell or tissue in a nutrient medium under aseptic conditions. The cell or tissue is placed in a nutrient medium where it forms a mass of cells called callus. This callus is then transferred to another nutrient medium where it differentiates and forms a new plant.